There is a G protein linked receptor and an inactive enzyme. They both are transmembrane proteins, meaning they are found on both sides of the cell membrane. A ligand or a signal protein heads toward the receptor to bind to it. After the signal protein binds with the receptor, a G protein with GDP attached to it attaches to the receptor. The G protein then picks up a GDP and it becomes activated. Next, it detaches from the receptor and binds with the inactive enzyme, which is adenylyl cyclase. When it binds to the enzyme, it changes its shape, making it active. Now, the adenylyl cyclase converts ATP into cyclic AMP, or CAMP. The CAMP then activates a protein kinase which goes on to phosphorylate additional relay proteins. Finally, the last protein in the cascade produces the cellular response. In this case, the cellular response is the release of calcium ions from the smooth ER. This is the tyrosine kinase receptor system. It starts with two transmembrane tyrosine kinase receptors. Two ligands or signal proteins appear and make their way toward the receptor. After the signal proteins bind to the receptor, the two tyrosine kinase receptors become dimers. Then 6-ATP loses a phosphate group and the 6-ATP turn into 6-ADP molecules. The 6-phosphate groups bind to the tyrosine receptor. Then two inactive relay proteins bind to the phosphates. There can be more than two inactive relay proteins, but this depends on the message from the signal protein. After the relay proteins bind to the phosphate, they become active relay proteins. They relay their message to the ER and create a cellular response. In this case, the response is the release of calcium ions from the ER. We show the effects of anthrax poisoning. In the normal pathway, an antigen of a harmful bacteria binds to a G protein receptor of a white blood cell. Then, a G protein with GDP attached to it attaches to the receptor. The protein then picks up a GTP and it becomes activated. After this happens, the G protein leaves the receptor and attaches to the adenylyl cyclase. After this, the enzyme changes shape and activates, catalyzing the transformation of ATP into CAMP. The CAMP then activates protein kinase, which phosphorylates other proteins. Finally, the cellular response happens. In this example, the cellular response is a change in the shape of the cell membrane so a white blood cell can perform phagocytosis and ingest the pathogenic bacteria.
The normal pathway in white blood cells is affected by anthrax. Anthrax poisoning occurs when inhaled bacteria emit a harmful toxin. Just like normal, the antigen binds to the receptor, changing its shape and triggering the G protein to bind to the receptor. Then, the G protein gets activated by switching out a GDP for a GTP. However, the toxins emitted from the bacteria change the shape of the adenylyl cyclase, which is the inactive enzyme. As a result, the activated G protein cannot bind to the adenylyl cyclase, and this inhibits the production of CAMP from ATP. Due to this, the white blood cell is inactivated and it cannot kill harmful bacteria. This makes the anthrax bacteria thrive in the organism, and if this is prolonged, the organism can die. Symptoms of anthrax poisoning include fever and chills, chest discomfort, shortness of breath, confusion or dizziness, cough, nausea, vomiting, stomach pains, headaches, sweats, extreme tiredness, and body aches. The standard treatment for anthrax is a 60-day course of an antibiotic such as ciprofloxacin, Cipro, or doxycycline like Monodox, Vibromycin, and others. Also, antitoxin therapies have been developed. Instead of going after the bacteria that causes the disease, these medications help eliminate the toxins caused by the infection.